My name is Najiha Nika Suraya binti Muhammad Arif Empati with metric number 111210.
Hi, my name is Hanifah Sharina binti Ilham. My matrix number is 110106 and I'm from group 6.
Hi everyone, my name is Moshita binti Muhammad Zaki and my matrix number is 110734.
Hi, my name is Kasma Cerida with metric number 110451.
Introduction. Reducing sugars have the property to reduce 3,5-DNS acid into 3-amino-5-NS acid in alkaline solution. The method quantitative analysis of reducing sugars by 3,5-DNS acid is based on the detection of presence of free carbonyl C double bond or group of reducing sugars. Initially, oxidation of the ketonic and aldehyde functional group of fructose and glucose respectively by 3,5-DNS acid to by 3-amino-5-NS acid in alkaline medium, which is from yellow color to orange-red. Objective: to introduce the students to the use of DNS method for quantifying sugars concentration in given sample.
The materials are DNS, reagent, Rochelle salt, cuvette, test tubes and test tubes rack, hot plate glass beakers, distilled water, micropipette and tips, and selected drinks. Methodology. First, 6 clean and dry test tubes were prepared. Next, 1 ml of DNS reagent was added into all test tubes containing samples as measured according to the above table. The test tubes were boiled for 5 to 7 minutes. The test tubes were let to cool down to room temperature after boiling. 1 ml of Rochelle salt solution was added into the mixture. The color development of the mixture was read at 575 nanometer by using a spectrophotometer. A blank test tube was used and set as negative control to eliminate background readings.
Based on the result obtained, the glucose concentration in the sample was determined using the DNS method and the standard glucose curve prepared in Practical 2A. First, a series of sample dilutions were prepared by mixing different volumes of the sample with distilled water. The dilution factor for each test tube was calculated using the ratio of the initial sample volume to the final total volume. After reacting the samples with DNS reagent and heating them, the color change from yellow to reddish brown was observed. The color intensity decreased as the sample became more dilute, indicating lower glucose concentration. Next, the absorbance of each sample was measured at 575 nanometers using a spectrophotometer. The absorbance values were then multiplied by their respective dilution factors to obtain corrected absorbance readings. The glucose concentration was calculated using the standard curve from Practical 2A. Using the linear equation y equals 1.4134x minus 0.0285, the glucose concentration in grams per liter was determined for each sample. The results showed that the undiluted sample had the highest glucose concentration, while the more diluted samples showed progressively lower concentrations. This confirms that absorbance is directly proportional to glucose concentration, which is consistent with Beer-Lambert's law. Overall, the results demonstrate that the DNS method is effective and reliable for determining reducing sugar concentration in unknown samples.
Discussion 1. Objective and principle. The experiment aimed to quantify reducing sugar concentration in varying sample dilutions using the DNS method. This technique is favored for its simplicity, speed, and ability to handle high sample throughput. 2. Methodology. The tubes were covered and boiled for 5 to 7 minutes to facilitate the reaction. Tubes were cooled to room temperature to ensure accurate spectrophotometric readings. Rochelle salt solution was added after cooling to stabilize the color intensity and prevent oxidation. Absorbance was measured using a spectrophotometer to calculate glucose concentration. 3. Reaction mechanism and results. Chemical reaction. Heat and alkaline conditions convert the yellow 3,5-DNS acid into the reddish-brown 3-amino-5-NS acid. in the presence of glucose. Concentration trend. The results show concentration ranging from 1.049 gram per liter down to 0.020 gram per liter. Observations. High concentration. Darker reddish brown color, which means more DNSA converted to ANSA. High dilution. Lighter color intensity and lower absorbance reading due to fewer glucose molecules available to react with the reagent. 4. Experimental considerations and errors. Dilution impact. Sample dilution significantly impacts results. Adding water reduces the concentration of glucose molecules, lowering absorbance. Safety. Caution is required during the high temperatures boiling step. Sources of error. Accuracy is critical. Common errors include air bubbles in micropipette tips or incorrect volume of reagents or samples, both of which lead to inaccurate quantification.
From experiment 2b, we can conclude that in this experiment, 3,5-dinitrosalicylic acid (DNS) method is used to determine the reducing sugar of the sample. The absorbance spectrums of unknown glucose concentration had also been determined. From the result, we can know that the glucose concentration can be determined using the glucose standard graph that we obtained. The investigated parameters like linearity, limits of detections, limit of quantification, precision, and accuracy confirm that this method is adequate, reliable, and suitable for the study of reducing sugar in this experiment. Thank you!